So, as we go through these next few slides to wrap up this lecture, basically we got a little bit cut off, but in other words, each ion's Nernst potential combines to create a total resting potential of negative 70 millivolts. Then when an action potential comes, then we depolarize the cell and cause another action potential. So resting potential is the resting electrical state of the cell at a particular time. So no movement of ions across the cell membrane at a specific point in time. So a change in the cell potential that actually occurs over a period of time would be an action potential, like a pulse wave. So let's kind of take some examples and put this a little bit into perspective. So imagine you can apply an electrical stimulus to a neuron cell membrane. What happens? The area closest to the stimulation shows large change in potential, right? So something that's stimulated close, obviously, is going to have a large change in potential. So the change in potential decays as it moves further away from the location of stimulation. If we increase the intensity of the stimulation, it increases the distance that the potential will travel. Again, that goes to that temporal and spatial summation. So think of a drop in a pond. Ripple decays as it moves away. Okay, something very similar happens when you stimulate a neuron. So summation, if we have multiple ripples that add together, we make a larger wave. So an action potential for a particular neuron, imagine we can apply a current to make the voltage in the cell more negative or more positive. Negative is going to be hyperpolarization, positive is going to be depolarization. So depolarization and hyperpolarization would spread like a ripple along the membrane. So still taking that analogy, now imagine that there is a wave machine under the surface of the water. If the initial ripples are big enough, they trigger the wave machine to create one giant wave. Okay, something similar occurs in a neuron when an action potential is generated. Again, if that little ripple or some of those EPSPs, right, hit a certain threshold, we can now trigger an action potential or trigger that big wave. So how does this happen? Sodium and potassium channels are open under certain membrane voltages and closed under others. So sodium channels open when the potential depolarizes from negative 70 to approximately negative 45 millivolts. Potassium channels open when the action potential reaches a peak voltage around positive 40 millivolts. So sodium channels in general go from deactivated, activated to inactivated. So another analogy to kind of put some things into perspective, think of the ion channels as being guarded by tiny little monkeys. So a small depolarizing current puts a few of the monkeys to sleep. Okay, this allows some ions to cross the membrane, mainly sodium at this point. These ions generate more current, which in turn puts more monkeys to sleep. Eventually, all of the monkeys fall asleep, pass out, all the channels open, and the potential reaches its maximum at around positive 40 millivolts. So the initial current must be equal to some threshold to put the first monkey to sleep. Physiologically speaking, the initial current is the summation of the presynaptic inputs. These inputs travel along the dendrites to the soma. Okay, it must be enough to activate the first sodium channels. And the idea of positive feedback is that a small initial current builds into a large current. So the first channel opens, which allows more ions to move in, which generates more current, which then feedbacks the system to open more and more channels. Again, we have this all or none principle, and I'll kind of make this a little bit longer for you guys to see, or I'll move it, saying that an action potential only occurs if the initial current crosses threshold or activates the first sodium channels. Go right there. So following the action potential, we're going to have this brief, so we can't have two action potentials without some type of kind of rest in between, and that's this refractory period. So it's this brief period immediately following an action potential in which no second action potential could occur. So all the monkeys are completely awake, which allows for hyperpolarization, meaning there's going to be no action potential. Then we have the relative refractory period, which is a brief period following the absolute refractory period. Can generate a second action potential, but current threshold is much greater. So here you guys can kind of see what happens to our membrane potential, okay, as it gets depolarized, thus causing an action potential. So we see that the sodium channels open, okay, while the potassium channels. So we have this influx of potassium, we have an efflux of sodium, and then eventually we have repolarization in which potassium starts to re-enter the cell, and then which leads to our refractory periods, in which it's a lot harder to trigger an action potential. Because if we have several action potentials happening in a row, then we're going to have something that would be, if you guys have ever seen like the freezing goats, okay, that's something that would happen to our bodies as well. And obviously the freezing goats, it's not very efficient to freeze every once in a while. So we have postsynaptic inhibition. We talked a little bit about this with the interneuron inhibition and the muscle spindle example. So the nervous system can resist the generation of an action potential through postsynaptic inhibition. How? The resting potential of the neuron is made more negative. Okay, so the inside of the cell is made more negative, making it harder to create an action potential. Okay, and these examples of these would be our Renshaw cells and our 1A interneurons. So our Renshaw cells are alpha motor neuron synapses on a Renshaw cell, which are excitatory. So the Renshaw cell, think of it as basically taking feedback from a system and saying, no, you do not need to contract. So they receive excitatory collateral from the alpha motor neuron's axon as they emerge from the motor root, and they're kept informed of how vigorously that neuron is firing. They can then send an inhibitory axon to synapse with the cell body if the initial alpha neuron and or alpha motor neuron of the same pool. So they basically say, okay, there's way too much going on here. We should inhibit some of this excitation that is occurring. So why does this occur? I mentioned the negative feedback, counteract any incidental activity in muscles, and can be controlled by the CNS to be more or less inhibitory. Again, the feedback gain. Then we have the 1IA interneurons. These receive signals from the muscle spindle afferents, send axons to alpha motor neurons that control antagonist muscle. Okay, and again, these axons already exist, but we're kind of saying, okay, these axons go here. So we'll see the reciprocal inhibition of that stretch reflex inhibits the action of the antagonistic muscle. We have presynaptic inhibition. So postsynaptically, we're going to resist action potential from all inputs. Okay, presynaptic inhibition, the postsynaptic cell can resist input from a particular presynaptic cell, again, kind of modulating that stretch reflex. So the postsynaptic cell axon branches and synapses on one of its presynaptic cells, makes the resting potential of the presynaptic cell less negative. The amount of neurotransmitters released is related to the change in potential from resting to action potential, and then this change thus is therefore reduced, and the presynaptic cell releases less neurotransmitters. So to go into a little bit of more science about where we're at with some research, we have in creating neurons to cause human movement, we can actually, there's a lot of stem cell research out there, and we'll briefly talk a little bit about this. Stem cells are cells that divide to produce more cells. Okay, so they're classified based upon potential to become different types of cells. So they can become any cell in the body, most cells in the body, few cells, so on and so forth, and can divide and replenish themselves. So stem cells in motor control, neural tissue has very limited ability to regenerate. So stem cells might be useful for replacing damaged cells in people with brain injury, spinal cord injury, Parkinson's disease, so on and so forth. as they can grow and replicate. So this may be useful for growth factors as they generate. But we do need to consider how to get stem cells to the correct location, okay, to help repair some of that damage, how to get them to become neurons and then connect to one another to have that communication, how to train the nervous system to use the new connections, how to keep stem cells under control, okay, to make sure there's no tumor growth. And then we also have, obviously, the ethical considerations of where we get our stem cells from. And that's the end of this lecture.